today, Detlev Schild. Detlev is coming from Goettingen. He got his diploma in physics from the University of Goettingen in 1979, and his MD from the same university in 1982, and a PhD in 1987. In 1997, he was appointed head of the Department of Molecular Neurophysiology in the Center of Physiology and Pathophysiology at the Medical School of the University of Goettingen. And since 2012, he is the head of the Excellence Cluster Nanoscale Microscopy and Molecular Physiology of the Brain. He's interested in understanding how smell works, and his lab is using a multitude of techniques, including electrophysiology, single molecule detection, photochemical and high resolution imaging, as well as computational techniques. And today he will tell us how we can understand the code of smells by looking at activity correlation.
Yeah, thank you very much for this kind introduction. I'm very happy to be here actually, and as you might see, I'm rather burnt. It is also nice weather. Last 2 weeks we were squiddling around in various parts of this island. I always wanted to go to and to see more closely, and I eventually did. And fortunately, as I said, we had a marvelous time, and I didn't manage to put the nice background of Kenya or whatever on this. You know it all better than I do, so it doesn't make a lot of difference. Instead, I put this thing here. Is there a pointer anywhere? I put this thing here, which is a glomerulum, and that's the neuropil. So glomos in Korea has a Greek origin, I guess. Glomos in Latin at least means a skein, right? So a skein of wool practically is a glomos, and a little glomos is a glomerulum. And the glomerulum is the place where the primary fibers from our neurons of the sense of smell go where they head to, and then they make contact, make the first synapse in these glomeruli of the olfactory bulb. And that's our main interest. How does that work? How's the projection from the sensory cells to the primary cells in the brain?
So it's the projection of the cell body.
This is, here come projection of the cell bodies into the glomerulum, and then mitral cells in the olfactory bulb pick up the information.
The cell bodies are projected to start.
I will show you in much more detail, yes. Now you see how it works in humans. Just to show you the hopeless case first. We have 2 square centimeters of olfactory mucosa here. And here you see in more detail how it is. Here's the olfactory mucosa with the sensory cells in the olfactory epithelium. Then we have the Gregory-form plate here, which is the part of our skull. Here is the Gregory-form plate, as that's what it's called. And there the axons of the receptor cells pass through that plate, through little tiny holes. People thought about almost 200 years ago, then when you have a cold, whatever gets out of your nose comes from the brain directly. That's not really true, but that's an idea at least. And then those fibers enter the olfactory bulb. You might not see from the last row, but here are those glomeruli. And then you have mitral cells and the projections all over the brain. And here is another zoom in this olfactory mucosa where you have the sensory cells and the cilia. And the cilia are in the mucus of the nose, and that's where the ligands, where the odorous molecules make contact and bind to olfactory receptors as one of the first steps. Now, this slide is taken from Linda Buck from her Nobel Prize talk. You know that she and Richard Axel won the Nobel Prize for the discovery for her discovery, I should say, and he made politics of olfactory receptors. And this is from her talk. I don't know who this is, but I thought it would be in this country adequate to put this fellow there, because that is actually the first person who claimed that molecules, he called them atoms of course, but that molecules, we would call them molecules nowadays, must be different if they have different actions in the body. And from this fellow, unfortunately, we have very little left. There are just a couple of fragments. But in De Rerum Natura of Lucretius, he said, for instance, thus simple it is to see that whatever can touch the senses pleasingly are made of smooth and rounded elements, for instance this one here, while those which seem the bitter and the sharp are held in twine by elements more crooked, maybe this one here, grapefruit, differently. So the idea that there are different kinds of atoms, of molecules, is very old, it's 2,500 years old, and lived on for about 500 years, and then everything was smashed, and people got other stuff, and then very lately, people came around with that idea again in chemistry, thinking about that gap in between keeps you weeping for a while. Okay. So as I said, this is the hopeless case. Why is it hopeless? Because you cannot do experiments in that. You can only do black box experiments in humans. So you can apply odor, you can give an odor, I can give you a grapefruit or something, I can record from your skull, but the whole thing is a black box, like a computer. You have an input, you have an output, whatever's going in between, we just don't know. There are whole black box faculties like psychology, which is okay and totally justified. You just have to admit, I don't know what's going inside, you know? And that's the difference, one of the major differences between physics and biology. In physics, it stepped down until 1900, until the atomic physics, quantum physics, and so on. And then you synthesize things and build transistors, build semiconductors, and so on. And then you have these NAND cutters and computer chips and so on, and you know what's going on inside. So it's no longer a black box. And then you can say, this is on my input-output relationship, and that's it. Now, the input-output can be done, but we cannot tell anything in humans about what's going on in between. And that's why it's impossible to do this kind of experiments in humans. And why is that so? There's two reasons in particular. There are many reasons actually, but two major reasons. One reason is this cribriform plate. Even if you were allowed to do experiments in human, and of course you are not, but you could take mice, okay, they also have a cribriform plate. Just try to make a preparation where you have the sensory cells and part of the brain within the same dish and do experiments on them. It's just not possible. You need a butcher and their instruments, but it just doesn't work even then if they are macro. It just doesn't work. And if you think about it, that's very customary. We are so used to it. We are so used to it that there is no prep within the hair cells and the cochlea, what is it, the anterior, posterior, ventral, dorsal, cochlea nuclei, and so on. There is no prep where you have everything in the same dish. You cannot do experiments applying sound and see what's going on in the brain. Okay, so that was exactly our goal when we started this project on olfaction, and it took us 9 months to find this animal here. Even in the adult form, so in the frog, you have a cribriform plate, but you don't in the tadpole. There's no. Here's the eyes, obviously. That's the noses, the nostrils. And there's no bone in between. And that means, and the system works. So it's a functional olfactory system. They recognize themselves, they search food, there's predator avoidance, and you can make, and that is most important of it, the nose-brain preparation. And all the stages, the receptor cells and the brain stages are accessible experimentally. So you can do, say, Ashland recordings in the receptor cells or in mitral cells if you want. You can do imaging in those and the others and so on. That was very nice. And they're fairly transparent, which is important for all kinds of imaging, of course. So what you can do then, is you can just cut out this piece here and have your nose brain pressed. And wherever you cut, you can cut here or there. You get as much as you want of the brain in here. And here you see the olfactory nerves. These are chromophore cells here. These are the olfactory mucosae. The stimulus arrives here. Here the receptor cells in there. And this is the olfactory bulb. Here's the accessory olfactory bulb. But you cannot see all that here, of course, in this simple image. But the nice thing is, you can do very nice experiments now. You can just blow a little bit of amino acids, that's also very nice. All higher vertebrates, those thinking beasts like ourselves, a little bit thinking at least, smell stuff which are incredibly difficult to manage. They're not hydrophilic, they're lipophilic, so they stick to glass, they stick to plastic, you never know what kind of concentration you apply, and so on. That's much, much simpler in these beasts because many of the stimuli are, for instance, just amino acids, or bile acids, or say alcohols, or that kind of thing, which you can easily dissolve in water. So experiments are very easy to be carried out. And you can patch them the olfactory mucosae, and you can also patch them and image the olfactory bulb, and that's what I'm going to tell you something about. Let's first have a look at stage 1 at the receptor cells, at the structure and the activity of receptor cells. First thing, how do they look like? These yellow things here are the cell bodies, the somata of the receptor cells. These long things are the dendrites. They have just one dendrite, probably the simplest neuron around in the whole body, just one dendrite. And here's the knobs. Here, you see all these knobs here. That's all olfactory knobs. That's the endings of the dendrite. And from that knob, we have the cilia issuing into the mucus, and now the stimulus come up here, bind to receptors here. And I'm not talking about those processes of construction processes and all that, all very interesting, but I don't use the word on that because that would be beyond the scope today. This is actually my first recording when I was allowed to do patch experiments myself. It was the first, so it's very old. And you see a depolarization here. This is a patch lamp in the current clamp mode. You see the receptor potential, and you see a couple of spikes here. Not untypically, two spikes very close to each other, and then it goes down, and that is it. This is the actual patch pipette at the real cell. So those days they were dissociated cells. You can record from dissociated cells. We left that because there are many disadvantages. And these are two sketches of pipettes. These are not real pipettes, but just sketches. And you see where you can apply and sample and do odors or pharmaca or whatever you want. Putting them in solution is not a good idea. I mean the whole community working on receptor cells, tried for 10 years to use odorants at reasonable concentrations, and it just didn't work. It just didn't work, except you use very high concentrations, and they're lipophilic sometimes, and then they just break holes into the membrane that gives you a depolarizing current, so the cell is excited, but it's not a specific response, so it just didn't work. Much later, just 5 years ago or 7 years ago, we were able to publish 2 papers on the endocannabinoid system of these cells. They have CD1 receptors, and the glial cells and the receptor cells themselves produce endocannabinoids, and those are necessary to smell. Whenever you block the CD1 receptors, so the endocannabinoid receptors, whenever you block them, you don't smell anything. So we need the endocannabinoids. And the endocannabinoids, that's the second of our papers there, are hunger dependent, interestingly, which is also interesting in itself. And I mean, all of you know that when you drive home on a bike, say, which is not the usual case, maybe you can be a little bit outside, but you're really hungry, you can tell what are they cooking in this house, what are they cooking in that house. When you're hungry, you smell everything, right? And that may be the sense of the endocannabinoid system as a whole, because it has been shown by fishermen, it has been shown in the auditory system. Whenever people are hungry, they hear better, they smell better, they see better, and so on. And if you see addiction and hunger might have something to do more than we thought previously. But that's also a story. I will not lose a word on today. But if you do this kind of experiment here, you obviously go into whole cell mode. You break this little patch here, and that means there's diffusion of solution into the cell and out of the cell, and that means after a while it doesn't work, there's too much washout. Therefore, if you want to record over longer times, what you have to do is just go with the pipette onto the surface of the cell, so-called on-cell recordings, and then look which kind of stimulus works. These are 2 different cells. This is one cell, that's another cell. This reacts onto L-methionine, so that's an amino acid, and it does not work on the activation of the CMP pathway by phosphorin. So what you see here is just spontaneous activity, and this is a stimulus-induced response. Well, this we have to consider then as a background. The spontaneous activity is a background to the olfactory system. That's not completely uninteresting to consider, because if you think that there are thousands and millions of receptor cells, all producing one spike per second, and if those fellows converge onto glomeruli, you have 1,000 spikes per second running there, so one every millisecond. If they converge onto, say, 5 or 10 dendrites at the outlet of the glomerulus, that would be a fairly high activity. And does that do anything to the animal? It shouldn't do anything to the perception because it's just background, it's just spontaneous activity. And therefore it's interesting to see what's the statistics of the spontaneous activity, and therefore we calculated the so-called autocorrelogram, so the autocorrelation function of spontaneous activity, and that looks typically like that. So you take the spike train, okay, you shift it with respect to itself and multiply, okay? And whenever you get an overlap, you get, for that lag, the component into the autocorrelate. And what you see is there's just a constant which corresponds to the spontaneous activity, which corresponds to 0. If you would calculate the autocovariance, it would be 0. So if you subtract the average, the activity minus the average would be 0 here. But in the beginning, you see a little bit of a positive correlation. And that means the cells have a tendency to spike with a very short latency. Spontaneously, that happens very rarely, but that's an interesting indication whether that might happen and be important in stimulus-induced responses. It's not necessary to do electrophysiology because the advantage is clear. You can measure all kinds of transduction processes, ionic channels, and so on. But with just one cell, okay, and that's not very bad. You might want to have many more cells. And that's possible with calcium imaging. And these are calcium traces, and whenever there is a jump here, like here, you might see this calcium increase may be generated by a spike, by action potential. And then by simultaneous calcium imaging and one-cell recording with PET, you can really verify that that's the case. That means that you can really do in the mucosa a kind of calcium imaging for which you can conclude the activities of the cells. But the bad thing is the whole mucosa is like a half sphere, like more than a half sphere, like a cup. Okay, so whatever your section is, you just get a couple of cells and that's it. So that's not very good. Let's go to the second structure then of this system, the olfactory bulb, the glomerular and the olfactory bulb. So this stage here. There you see one zone here where you see the glomerular. Now they appear white here. Here are the mitral cells, mitral cell bodies only, and here are the granule cells. Okay. This is fairly old data. Here it's stained. It's the same kind of thing. Olfactory nerve fibers in green entering the bulb. This is the accessory olfactory bulb. Here entering the main olfactory bulb. And these red things is synaptophysin, so that shows you where the synapses are. Any kind of synapse in this part of the brain. And here you see the glomerula. So the fibers enter the glomerula here. And the mitral cells then have dendrites which pick up the information from the glomerula. How do they look like? Mitral cells have a couple of dendrites. It was claimed for a while that there's just one dendrite from one glomerulum, but that's not true. In particular in frogs, it's not true, but not even in mice. And here you see this mitral cell picks up its information from 3 different glomerula. And the interneurons in this olfactory bulb come from the granule cell layer. They are the granule cells, and those make contacts at their ends with mitral cell dendrites. So it's a fairly simple system. In higher vertebrates, you would have another type of interneurons here in this sheet, the so-called periglomerular cells, which we at this stage do not have. They come up later. Now what you see here is what's going on here. You know, yesterday I have to apologize. Yesterday I didn't block the software updates, which has the consequence that it's very weird. You saw before there were having trouble with the computer, and Now, this is a granule cell. They don't have axons, they just have dendrites when in contact with the mitral cells. And on the dendrites, you see some kind of appendages here. And that's here again, that's a zoom of the other cell. And they have appendages, and those are spines. And on those spines, you have NMDA and AMPA channels. And on the other side, on the mitral cell, you have GABA channels, GABA A channels, and GABA C channels. So that's the dendrodendritic synapses you're finding all over the place there. Without interneurons, it looks fairly similar. It looks fairly simple. The idea is that receptor cells of the same type, here drawn by the same color, enter the same glomerulum, which is not perfectly true. They sometimes bifurcate here and can go via another way into the same neuron, but they can also go into a neighboring glomerulum. That also exists, so this is simplified. That's one simplification. Other cells, other receptor neurons, focus onto another glomerulum, and so on. And then we have the interneurons here and making contact here. From between glomerulum and mitral cells, there's lots of modulation possible, and I'll come later back to that. So the whole system is rather, how should I say, in character combinatorial, in the sense that an odorant usually does not excite or just bind to one type of odorant receptor. Many ligands can bind to more than one odorant receptor. An olfactory sensory neuron does not just have one receptor all the time. So that's the dogma, but that has been shown in many species that that's not really true. Many neurons, especially in development, can have more than one receptor, many more sometimes. Those sensory neurons then project to glomerula. That's the black thing here. Ligand, receptor, receptor neuron, glomerulum, mitral cell. But it's not that simple, because you've seen here just already two exceptions to that. Now, as I said, the olfactory receptor cells can also, of different kinds, converge to the same glomerula, which is also one factor of calculating combinations. And then the mitral cell does not only pick up the information from one glomerulum, but it can pick up information from many glomeruli. So at every stage, you have the possibility of getting combinations. Just looking at the first stage, you can imagine like this. These are the concentrations of L possible ligands in the system, okay? Each ligand occurs at a certain concentration, and that ligand at this concentration can then bind to various receptors into the same of the same cell, and in the end generate this activity R1. Okay, and the rest, and for the other cells that's likewise. But it seems linear algebra, because the notation is taken from linear algebra, it is not, and not at all. It's much worse, because the B's are dose response curves. Okay, so they are nonlinear sigmoidal curves with a threshold and with a midpoint and so on. And the R's are not just numbers, but they are activities, so they are delta cones. They are just spikes. Okay, that makes the whole thing much more complicated, obviously. Now the problem is, if many mitral cells pick up their information from the glomerula inside in parallel and simultaneously, the only way of getting that information is measuring for many mitral cells at the same time. And that is, of course, not easy. The traditional way is just poking around with a pipette and measuring from one cell. That's what people did for many years, for decades. And I think much of our textbook knowledge is streamlined that way. We think in streams of single channels, actually, and very rarely do we think in ensembles of many cells at the same time, which then go on in time. And so the challenge is to measure many mitral cells at the same time. And we try to do that in the following way. We just took a piece of the olfactory bulb, take a soma of a mitral cell, and try to record and try to see optically the whole cell. Okay, so we took the function in the soma. You have to imagine this is not just an image, okay? We take this as a time sequence. This image is taken 10 times per second or something like that. So it's actually moving, okay? That means to every region of interest, we have a time function, which is a time function in this region of interest, okay? What we can do now is, we can go around the whole image and see where do we find something similar, something highly correlated to this function. And you find such places. For instance, these 3 places here. And you see it looks different, obviously. This is the soma. You can average over many pixels, so you get a fairly smooth signal. But nonetheless, you see here's 3 peaks, there's 3 peaks, there's nothing going on here, there's more 4 or 5 peaks here, and here are 5 peaks, here are 5 peaks, and so on. So you have fairly high correlation coefficients, okay? So in order to plot the fluorescence of our calcium dye here, we could plot the correlation coefficient between this and that function and put it at that place. Okay? Is that clear? That's the basis for the
This is supposed to be different neurons that are correlated.
But that's not clear.
Or different parts of the same neuron, for example.
That's not clear. We just go around look for the same activity. Okay? That's an interesting question because you would suppose it's the same. But we'll come to that point later in a second. That's a very good question. We thought at the beginning it's just one neuron, but that's not the case actually. And if you plot the correlation coefficient between that and that at this point, you plot something which is not white. This would be white, right? So 255 in 8 bits. This is not 255, maybe it's 220 or 210, but it's a fairly high correlation. And what you see then is, you see the whole neuron in terms of correlation coefficients, which you did not see in terms of resonance intensities. That's the new point here, okay? Now, unfortunately, I can tell you this is a very rare case, very long, where 2 dendrites and 1 axon are almost in the same plane. Okay, that doesn't occur, obviously. That's just by chance it occurred once here. And usually it goes down here. And in fact, this is our visual system. You all know those triangles you look at, and you see a triangle, but if you really look at it, it's not there. Right? You remember that kind of thing? 2 triangles drawn into one into another, and the half of them is missing, and so on. That just means our cortex has reconstructive capacity to use. And of course we conclude that this is the same axon as this, but there's no proof of that because here it's missing, and that could be another axon, right? So we are just interpreting all the time. So what we need here, of course, is a measuring technique which does not measure just one plane, but a little bit into z direction as well. So dendrites lead the focus volume, and image stacks are badly needed here. And that's the reason why we built a laser scanning microscope. And the basics of a laser scanning microscope is in fact quite simple. So we don't have all the fancy stuff Zeiss has, just a couple of lenses and a scanner. But in between, we put a cylinder lens here. Here, there's the cylinder lens. And that means while normally you would have, here comes the laser, right? Just to explain briefly, here comes the laser. Here it's on the scanner, so there's the laser coming somewhere, right? Then you get the scanner, you get another mirror, and they move both. That means that the laser beam going here can be directed wherever you want by the scanners, and usually it's a round Gaussian-shaped intensity thing, which you can, with the lens, focus onto one spot, right? That's the idea, and then you go spot by spot and scan the whole image. That's not what we do here. We put a cylinder lens in here, which means that we form a slide, a line, so to speak. Here, it's a line, here, and that line is projected then onto the back focal plane of the objective. Now, if you think this is the objective here, say, like this, rather, okay, and you put a line in here, and you move that line like this, okay? What does happen? In this direction here of my finger, the extension is as broad as it can be, and that means that direction will be focused up to one point in the focus plane, okay? So any focus plane. In this direction, the line is just one point, and that means in this plane it would be broad as possible. And that means that line is projected onto an orthogonal line here. And while this line by the scanners make this movement, this line makes that movement. And that line scans the image, obviously, as fast as normally one point would scan one line. That means it's about 250 or 500 times faster than normal laser scanning. And in fact, the bottleneck is now how many photons do we get out of the preparation. The bottleneck is no more the speed of scanning. That's high enough now. Okay? And that way, that's now back projected. The fluorescence is back projected, taken up, in this case by a line CCD camera. Now we're using a normal camera. And that line then is reconstructed for all positions, and then the image is reconstructed. That's how it works. And if you do that, and now I hope this works, yes, now we take the image and we measure it in 18 planes, okay? This one plane, another plane, another Z-planes. Then we take one region of interest and measure the time function in that region of interest. And then we look for the same time function in the whole slice here, okay? And you get all those plots, but some are autofocus. But if you do the same thing in all 18 slices, you get the same thing. You can merge it and get a 2D projection of 1, 2, 3, 4 neurons and one glomerulus. The same for different activities. You stain the different activities differentially, merge the whole thing, and by that you get a couple of glomeruli with the mitral cells attached to it. Okay? And this way you use the activity as a marker for contrast. Okay? The different points in the tissue have different activities. It's just one dye. The different colors here are arbitrarily assigned. You could use any colors. It just means one color is different from the other one. Okay, that's all. All the rest is arbitrary. It just means that this mitral cell and that mitral cell and that one take up their information from the yellow glomerulus, so to speak. And so on. So what we use is by using just one dye, using the activity of the neuron itself, and reconstructing the structure of that glomerulum with the mitral cells attached to it. And then doing the whole thing for all the rest of the game. Okay? I can show you another one. That's the same thing. You go through the Z planes, stain all the different activities differently with different colors. Well, that's too complicated sometimes, then you stain just a couple of them. And you can also stain just one of them if you want to look at just one. And the red ones are, of course, just the nuclei. And that way you can reconstruct the whole 3D network. And that would be just normal morphology. And it may remind one or the other of you of the so-called rainbow technique they invented in Lausanne. A couple of years at the St. Swiss Bank in Lausanne, actually, a couple of years ago, where they put 3 different GFPs into cells, and as those GFPs were expressed differentially, you always get different RGB combinations, and thereby stain different cells differently. But that's just morphology, as this is just morphology up to this point. But remember, we still have the time information. And that means we could come now and stain this glomerulum with the attached neurons to it, more green, brighter green, if the fluorescence goes up, and we put it dark green or almost black if the fluorescence is down, and thereby coding the time course, right? We have the time information, and if we use it, you see something like that. You can stain them when they are active in time. And thereby you can really see how they communicate, how they talk to each other. And that is exactly what we're doing right now within single glomeruli to follow from the presynaptic to the postsynaptic side to see how the, say, 3 or 4 presynaptic signals are gathered and processed postsynaptically, which wouldn't be as easy otherwise. Let me show just a few applications of this. We call it activity correlation imaging or activity contrast imaging. Nothing to do with accusative infinitive. What you can do, for instance, you could just see, if you do this, you find the same activity. This is fluorescence intensity, and the correlation map tells you, that this cell is highly correlated with that. So it's the same time function here and there, okay? What you can do then, for instance, is you can just measure those activities, then come with patch by patch with 2 different dyes, stain those different neurons, and look how, how they branch, where they go to, and so on. And that's, of course, not completely uninteresting. And you see, now we go through the planes, both neurons are stained, we go through the planes, and you see at a certain point they meet. And only at this point they meet. They meet nowhere else, those dendrites. They have different ways to the bulb, but they only meet within the glomerulum. They innervate commonly. They innervate also other glomeruli, but this is the only thing. And within the glomerulum, then they go there. That's the first thing, and take up information. But what also happens is, now once you know them, you can patch clamp one of them, for instance, depolarize cell 1, okay, and image calcium in the other one, and you see a calcium change here. You can hyperpolarize it and see this one. And that is significant in the sense that the manipulation of one cell in the sense of depolarization or hyperpolarization has an effect of the other cell, which is correlated. And as we know, they don't meet anywhere in the bulb except in the glomeruli. There's necessarily an electrical connection in the glomeruli, and that in the glomeruli actually, and that means there must be gap junction. That had been known before. That's not our discovery. In fact, if you take the coupled cells, you get this here. You depolarize here, get the calcium increase in the cell. You hyperpolarize, get this calcium decrease. And if you take just any cell here around, you get just more or less nothing, this curve or that curve. And that's very helpful. It's extremely helpful, because just imagine you want to do double patch. You have one cell, which one should I take? When I have one cell, which one is the interesting other cell I might be interested in? You just poke around there and see whatever you want to see, but you won't see anything maybe. Or something very complicated, mediated by a couple of different cells in between. But the directly cell, this one here, are probably never get, maybe 1% at best of the cases. And that means you need a frustration threshold, as you might only have when you learn the violin starting at 4 or something. You need extremely high frustration threshold, so you wouldn't find anybody who would like to do that. So
this correlation maps you make by having tissue simply cultured and imaged without some kind of stimulus. Is that the basal activity of the cells?
Yes. That's the best way of doing it.
It simply shows you the connectivity.
Exactly. That's the easiest way and the best way of doing it. That's an interesting question, because we formerly trapped into that trapdoor. Because if you don't do so, we feel that's stimulating. Stimulation is always okay. If you do that, no. If you stimulate, you synchronize lots of cells. And then they seem to be the same cell, but they are not actually. While in the spontaneous case, the probability that they're different cells is much higher. And the longer your record is, you could ask what's the resolution now to classify two cells as different. That depends on the length of your record. For a very short time, they might look somehow correlated. But the longer you go, if they're still correlated after 10 seconds, that would be extremely rare. Okay. Now, we knew from morphological work, not of ours, that they were connected by gap junctions there. And in fact, here's the same effect. Hyperpolarization, now you have double patch here, not patch and calcium imaging as before, but hyperpolarization, and the other one is also patched now, and you see an outward current. Then this cell is hyperpolarized, and you see the outward current here. So those are coupled by gap junctions, we thought, and that was very plausible. And if you put a blocker of the gap junctions in there, this effect is gone then. Okay, while you cannot block it by cadmium, which would block chemical synapses, and that doesn't change anything at the gap junctions. Okay, so that's the proof that those different two mitral cells, which meet nowhere except in the glomerulum, which they commonly innervate, are coupled by gap junctions. Which is interesting in itself, because if you put a dye only in one cell, you don't see that dye in the other one. Many gap junctions experiments, if you look in textbooks, okay, where the epithelial cells, you inject the dye here, and slowly you see the dye in the connected cells. A very classical experiment we carried out, I don't know, decades ago. But that's not the case here. And that probably means that there are lots and lots and lots and lots of them, but very small conductance. And in parallel, they lead to this conductance. You could even you could even measure their conductance because you know this is 25 millivolts hyperpolarization, and you know this current and voltage have something to do with the conductance. So you can really calculate the conductance of the complex of all gap junctions together. Okay. Good. Now, why just stimulating by voltage or current? Why don't we stimulate by odors and measure the same thing? And if you do that once, twice, you see that the reaction is fairly similar. There's a little bit of variation, but not very much, which means that the optical measurement in these cells is quite reliable. And you don't see only excitatory, there are 3 amino acids and the mix of them. And it's not only excitatory in different ways. Here, excitation is more than here. Here it's broader, that's not so broad, and so on. So the signals are reproducible from here to here. The gray signals are the signal trials, and the red ones are the mean of the, not the mean, are the average of the signal trials. And you see they look fairly similar from here to here, but they look a little bit dissimilar from here to here to here. So different stimuli generate different signals in different mitral cells, and the mixture is different again. And you also see other cells which are inhibited at the same time with a different time courses again. And you would never see that with Bernstein, that's excluded. You just wouldn't find it, no chance. This is the last application I would like to mention with activity contrast imaging. This is the end of a glomerulum, which is not much larger. The brightness shows you the correlation coefficient again, and so you see the glomerulum itself and in the dendrite is fairly the same. There's another dendrite here. It's fairly the same correlation, except here. Here it starts to look strange. What's this here? Really looks strange. Now what we did is we didn't take, this was the reference signal in the glomerulum. Okay, we thought let's take this here as the reference signal. Okay? And if you take this one as the reference signal, that's very clear, that's 1 because all the pixels in the reference area are correlated almost 100%. And then into the dendrite, the correlation stays high for a while and then vanishes. The correlation goes really down to 0 almost. Okay? So here's a correlation with respect to the glomerulum until here. And then something changes, and then it goes down. And seen from the other side, it looks like this. And this is what we call local ACI. You just take the reference somewhere, and you see there's something going on here. This can only be probably, I wouldn't say, but most probably there's GABA A synapses here active, which decouple the voltage from here to there, which decouple the signal on different parts of the dendrite, so that you can really say there's some signals which are still present in the glomerulum, are not, under certain circumstances, not transmitted to the mitral cells, while in other cases that's the case. Okay. So that's something you would never see with a patch pipette yet, because to see that, you would have to put different patch pipettes all at the same time onto the same dendrite. It just doesn't work. Okay. Now the question is, if we look at many mitral cells at the same time, how are the odors actually coded in the mitral cell? Most people would have thought until a couple of years ago that it's activity coding or rate coding as they call it. And that means look at, say, 20 mitral cells, 50 mitral cells, whatever you like, they're all active somehow. I displayed it as a movie, and then you see, like in fMRI, you know, those nice movies, images, and you see different activities in different places, and that movie seems to reflect what is happening upon the application of an odor. Okay? And that may be totally wrong. The first time this came to my mind is when, I don't remember his name, a fellow from London, Lebanon actually. In London studied color vision. Gave a talk at a neurobiology meeting, and they recorded the presentation of colored objects in the visual system, and recorded that the information of color and shape was recorded at different times. Which means that what we would consider one thing in the brain is not reflected as one thing at the same time. but is reflected as one thing, but coded somehow in a complicated way over time. We usually don't think about that, and we don't think about it this way because we are too video game prime or something. I don't know. We are too prime by going through the environment and seeing the olive trees outside. That's always the same every morning. It's always the same movie, always occurring at the same time because they're just standing around there. They don't move all the time. But it's not good. That may be different. So we just didn't know. And there was one particular point here which made us look into this more closely. And that is, if you give a certain odor to a mouse, or even to ourselves, the reaction in humans can be measured in about half a second. And in mice in about 200 to 300 milliseconds. Okay? So the reaction, if aversive in particular, to an odor can be extremely fast. Okay? This is not to say that we can differentiate whether this is this or that kind of citrus fruit, for instance. But we would just say smells like, but we are unable to say which exactly. That would take much more time. Okay. Now, if you think about the activities I showed you, which are 1 per second, more or less, maybe one per second or 5 per second in the spontaneous case, and say about 10 per second in the stimulated case, 200 milliseconds, 300 milliseconds would mean just 3 spikes. How can you calculate? And that's just in the receptor cells. Then you go to the mitral cells. Then you go to all kinds of nuclei in the brain, right? And then you come back to the reaction and you innovate your muscles, right? How can that work within 3 milliseconds if the receptor cells already are that slow? Okay, that seems to be a paradox here. Right? And that's why we took the opposite point to the rate code point and said, why, if it's only how could it happen, or can it happen that it's just the first spike? The first spike matters. Anything else is just, also significant maybe for differentiation, for other things. But to say whether there's something or not, maybe also whether it's aversive or not, that's just considered the first spike. Okay? And that's why we looked why we used activity correlation imaging in mitral cells and just looked at the first spike. So what we did here, I don't show you the movies for that because it's not really necessary here. Just 4 regions of interest, 4 mitral cells, and you see the reaction. The calcium goes somehow up here on the application of arginine in this case. That's of course too slow. One limitation of all imaging experiments is of course breaching. Breaching is always saying you just kill the dry molecules, right? And therefore you shouldn't image too much per day, per lifetime, per whatever. But when it's interesting, you should image as fast as you can. And that's what we did for a short while. We image very slowly, spontaneous activity, and then when we apply the stimulus, we become 100 per second, something like that image there. Because we were interested in when does the reaction go on. Then you can plot it on the other time scale, on the same time scale actually, but now zoomed, so to speak. And what you see then is that the dark blue cell reacts first, and the light blue, and the green cell, and then the red cell. So they really react one after the other, and there is an order of reaction. Which cells reacts when differs? And the question then was, is that significant? Has that something to do with the coding of the odor? Now you can see that within the cells, there's a certain variation within cells. Sometimes you have an outlier, that happens, but usually it doesn't look too bad.
And this repeats? These are simply repeats of the same application. Well, what are the different blue curves?
Applications, yeah.
Different applications of the same one.
Applications, 4 applications. 4 applications in cell 1, and you can also plot it as applications. You see the first application here, the second application, and the 4th application, and you really see that in the 4th application, this outlier here, the blue curve comes before the dark blue curve, the sine curve almost. It becomes before that, while it's usually after the other one. Okay. Now, you can define the rank correlation. There's many kinds of rank correlation measures. But the easiest way of defining it is if correlation is 1, if the sequence dark blue, light blue, green, red is the same. That will mean they react in the succession, in the sequence 1, 2, 3, 4. And that would mean correlation 1, because that's identical. And if they are mixed as much as possible, correlation should be 0. Now, I mean, how could you mix them? Now, 2 and 1 are mixed, 3 and 4 are the same. So there's one mix here, okay? How many could there be? There could be, there's 4 of them, 4 cells, so it could be 3 times 4 over 2, okay? So there's 6 possibilities are there. One of them occurred, that means it's not 1, it's 1 minus 1/6. That's the correlation coefficient here. That's defined like this. That's the number of inversions over the maximum number of inversions. That's what this number means. It's a very easy thing. Now, you don't have to look at application 1 and 4. You can look at any applications. And of course, you could also consider many more cells. In this example, for instance, 4 cells are relatively few. In this example, we had 18 cells actually. But it doesn't become very delightful to look at 18 such traces, obviously. We did that, of course, not just to represent them. It's kind of, you don't recognize anything. But you can now see what's the correlation coefficient. It's 8.3 somewhere here, okay? And how does that depend on the number of cells you have used for your evaluation? Of course, that becomes better the more cells you get. And if you take 18 cells, the histogram looks like that. If you take just, say, 4 cells or 5 cells, the histogram looks like that, okay? So that's to include the additional parameter of how many cells have we used to get that value R, because otherwise it wouldn't be comparable. Now you can calculate the average of that and the standard deviation to have a measure. That's quite straightforward, I guess. What we do now is we use this measure, this rank correlation coefficient, for different numbers of cells and for different concentrations of the same stimulus. The stimulus is arginine here, so 1 amino acid, applied to the glucosa, but at different concentrations, 5, 25, and 125 micromolar. So if we compare trials or applications from 5 micromolar as compared to 5 micromolar, we get this. That's exactly what we had before. It's just different applications of the same stuff. The same for other concentrations. But if we now compare 5 micromolar to 25 micromolar and 125 micromolar, we might wonder whether this sequence, which cells fires first, might change. That would mean that the concentration would be coded by the sequence of cells being activated. But that does not seem to be the case. So this is rather constant. The mean is rather constant. So applications of identical concentrations result in highly similar onset patterns, and applications of different concentrations result also in highly similar onset patterns. Okay? So the latency pattern or the latency factor does not appear to code for different concentrations. And this was just one slice. This is the superposition. This is grouped now over 8 slices or over 8 olfactory bulbs, and you see the average is almost the same now. It doesn't change over concentrations. It's perfectly the same. Now, let's compare that to the situation where we apply different odorants, different stimuli, arginine, methionine, and phenylalanine. And then you see we have different values of rank correlation coefficient here, and of course methionine with methionine gives a value, and so on. That's exactly what I showed you before. But now if we compare arginine with methionine, that mean value is completely different. And if we compare it arginine to phenylalanine, that's completely different, and so on. That means applications of identical odors result in highly similar onset patterns. That's here on the diagram. And application of different odors result in highly dissimilar odor patterns. And that means that the latency patterns, that's the pattern after how many milliseconds this cell, as opposed to that cell, reacts upon an odor, changes significantly. This is for one slice, and if you take 6 slices, cool them, you still see the marked differences.
But it's the same cells measured for different odors?
Yes.
Exact same cells?
All exactly the same cell, yeah, obviously. Yeah, sure. So that's the result then of that paper. Applications of different odors result in highly dissimilar onset patterns. And that's a strange way of thinking of. If you think about that, it really means that there's different times in different cells that matter to represent the same thing when applied at the same time. And that is quite strange a thought at first glance. But on the other hand, that increases the transinformation from the cell to cell signaling incredibly. Because now, depending on what the time resolution of the next receiving cell is, whether that can have a millisecond or not, instead of computing averages, can differentiate between milliseconds. That means a much higher computational power. Okay. Now, what after the mitral cells? What happens with these mitral cells? They fire at a certain point, and what then? They leave their axons somewhere in the brain, and then what happens? And very nicely, there was a paper from the Mori group. First author is Igarashi, but Mori is the fellow who's running that lab in Japan. And this is one of the most, for me, one of the most, how should I say, overwhelming papers I've ever seen. They wanted to have that published in Nature, but of course it didn't work out. There's too much. You know, it wasn't in this country, no, it was in Italy when Gregor the Great, Pope Gregor the Great, reordinated the vices and the virtues. Virtues are not so interesting for the church. Vices are much more important. People are more frightened by vices and virtues. So that he reordered them and put at the capital, so at the head of many others, at first capital vice was pride. Right? As your ancient fellows here say, hubris, right? I don't know how you call it today. Hubris. Hubris, right?
Hubris.
Hubris. So that was the first. Pride, hubris, right? That's all a neoplay and whatever you want, right? But the second was envy. And that's probably the reason why Mori didn't get it published in Nature or in Science. And so they published it in the Journal of Neuroscience then. This is just by chance process. And what is done here? This is obviously quite chaos, right? What they have done here is one cell body, and that's this one here. One cell body in the mitral cell is stained. Just one cell body by the person died. And then they looked at the propagation of that axon to the rest of the brain. And everything which is yellow here is that axon of one mitral cell. If you want to have something which may convince you to better become a sailor for the rest of your life or a postman or something, have a look at that paper, right? It is really hopeless, you know. And now, all of these mitral cells respond at different times. And they have, of course, different distances to all the nuclei this single cell, one single cell goes to. And we can't even say it's a mess. It is certainly not a mess. It is highly interesting. Okay. But it's also highly complicated. First class.
Is this mouse? Is this mouse?
This is mouse.
This is mouse, not frog.
No, this is mouse. This is a thinking animal, so to speak. And that's really something we have to digest somehow. So that's the reason why I don't go into mice and why I don't go into projections of mitral cells. And I would even say anyone, that's one of the citations in the book you have now, anyone who considers current experimental methods of understanding mitral cells action is of course in a state of sin. That is impossible because present techniques are just not sufficient to look at all those places here at the same time and see what's going on. Because the real picture would be much more complicated, obviously. Say something is projecting to the, say here, to a neuron here, but that neuron may project to here too, okay, and so on. And that is, fascinating on the one hand. And that was it, yeah. And the summary then is, odors are coded by large ensemble of receptor neurons, the code being a rate code. That's what we think at least. It may turn out to be more complicated. The structure and activities of neuronal networks like the olfactory bulb can be viewed in 3D by activity correlation or activity contrast imaging. And odor quality is coded by latency patterns of mitral cells or in mitral cells. And the players here Stefan Junek, who did a PhD in my laboratory, he's now at the Max Planck Institute in Frankfurt for Biophysics. Mia Alevka is still in my lab, and Chaiwen and Beijun moved to Genelia Farm and are now moved to Taipei, back to their homeland, Taiwan. And I would like to thank you for your attention. Maybe I'm a little bit over time. Sorry.
May I start with the first question? How different are these activity maps between the cells that have different colors? Is it easy to find a threshold to put different colors into different neurons?
Yes, as I said, that's a question of the resolution. If you measure long enough, it is easy.
And how long is long enough?
How long? It's just 2 seconds, maybe. 2 seconds, 10 seconds, depends on the activities. The longer you measure spontaneous activity, the better is the resolution of which cell is which cell. Obviously, those cells, you have seen it in the movie, it's not individual cells. If they are connected by gap junctions, then they have the same, more or less the same activity, highly correlated activity, and then you say two somas are with the same activity. So that was your question, actually, in the beginning.
And would this, you think this kind of pattern would also be seen in the cortex?
Yes, certainly, yeah. There's a PhD student in the lab who's doing 3 years now with me, and then is going to Amsterdam, and they want to look to the Fragile X syndrome, mice, and do the same thing in the cortex.
And they do like these distinct groups of neurons that are somewhere.
They are trying that right now, but I guess so. It depends a lot on the imaging quality. And the drawback here, I didn't show you that. There's obviously one drawback here. And that is you have to have the 3D steps. And you don't get them with the normal laser scanning microscope, because that does say more or less 1 image per second, and that's a little bit too slow. Okay, that's the reason why we do this fast laser scanning microscope, the light illumination scan as we call it. Size cells, a thing like that, they call it life. And they naturally withdrew it from many labs because it didn't work. Because they have, in the beginning, because they have a real-time processor processing all the data. There's obviously lots of data. The camera range is usually 1.5 megahertz or something like that. In this case, that's 70 megahertz, so lots of data to process and shift around. And that didn't work because they want their Windows machine, which everybody can use and is used to, and so on. And then they had a problem how to get the data in the real-time processing operating system to the Windows machine fast enough to show them. That was a major problem. They even showed up at our place and wanted to know how we do it. And they would say, well, we just use this Linux thing, which mostly doesn't work, as it almost wouldn't have today. And what we do then is we tell the Linux, you get 1 gigabyte of memory, of RAM, okay? And then we have a little program running up to that gigabyte, and when the first error comes, we know, and we put 4 gigabytes in there. And then the first error comes, that means we are at the threshold between 1 gigabyte and the second gigabyte. And then we use gigabyte 2, 3, and 4 as our private buffer to put data in there, and read it out and push it. So we do it with one machine only, and much faster, obviously, because it's all on RAM and not between 2 machines. Unfortunately, we are not allowed to. We are fight with them. So that's just a technical thing, which is not so important.
So how deep do you scan?
That also depends very much on the craft you have, and that's why I like catboats. Catboats you can scan as much as you want, as deep as you want. Usually, it depends. This you can image for about 100 micrometers or so, which for us is enough. It depends also. There's various kinds of chromophores. We also have albinos. There we can go up to 400 micrometers. But if you have in mouse, it would be more difficult. I guess with this machine you would go down to 35, 40 micrometers only. Because you have glia cells. This is I really look for a very simple olfactory system. We don't have glia surrounding the glomerula, as as there are in mice. You have the glomeruli surrounded by glia. There was even a glutamate spillover theory. So people thought glutamate goes somewhere else, you know, other synthesis. But we don't have glia, we don't have periglomerular cells. There's no myelin virtually involved. That means it's very transparent. That's of course crucial. That's as it always was. Galileo didn't see the moons before having the right telescope. So we need the right instruments to see something very simple.
And you're also working on the connectome to get. I mean, you have I assume pretty large chunk of the olfactory bulb. Could you try to work out the connectivities on the large scale?
Could try that, but we haven't started.
Okay.
As I said, our primary interest now is going into glomeruli. Actually, 3 people now going from mitral cells, the other receptor cells, and development looking at pre-post-synaptic transmission. Something weird is going on there. I don't know to understand how. And glomeruli are so widely distributed, even in insects. Maybe some of you have seen the recent book by Nick Strauss-Felder. He made a wonderful book on crustacean brains. A wonderful book with colors and it's fascinating. And they have glomeruli too. And from sensor cells converge onto glomeruli. And we have glomeruli in the spinal cord, in the posterior horn, during development, and then they vanish. We have glomeruli in the thalamus. We'll have in the triad state, we have glomeruli in the cerebellum, between granule cells and the efferents. So the question is, what really is a glomeruli? That is a fairly general question. Hopefully, something will come up. So that's the reason why we don't go into the overall connectivity, which would be maybe even a bigger project. And this is really where the computational ideas come in, because if you have some ideas how they are, how some of them are connected, you might be able to extrapolate that and to assume the others may be connected similarly, and then model them.